Good evening, everyone. My name is Joy Johnson, and we are going to be having people join us online. So I'm just going to let our online participants as well join. And I want to say a special welcome to them as well. It's great to see you all here this evening at Harbour Centre. And I'm really excited about this evening's lecture. So I'm the president of Simon Fraser University. And just really want to welcome you. This is part of an ongoing series, and I'm hearing an echo in the room somewhere. That's okay. No worries, no worries. I want to just welcome you all here. This is a part of a series of lectures that we're doing, and the big focus is making a difference for British Columbia. And I want to begin by acknowledging that we're privileged to be gathered today on the unceded traditional territories of the Coast Salish people, the Musqueam, the Squamish, and the Tsleil-Waututh. And we're really so fortunate to have this Vancouver campus on these territories. And I just, at this time, want to pay my respect to elders past and present. So we're here to hear from Dr. Colijn, who's going to showcase how data and mathematics can be used to prevent pandemics in the future. And I'm really looking forward to this lecture this evening. This is a time-honored tradition to have the President's faculty lectures. It's an opportunity to really showcase our amazing researchers and have them share their work with the public and members of our SFU community. I want to really thank the team at SFU Public Square. Thank you for all the work that you're doing to make us look good and to have the sound work and all the details taken care of. So this is an opportunity to promote the work, as I said, of our faculty, but also to have a dialogue. So there'll be an opportunity for you to ask questions this evening as well. As a researcher myself, I have to say I know how very important it is to think about the ways in which our research can be made accessible to the community so that people can understand the work we do, but also the impacts of our work. And that's one thing I really do love about Simon Fraser University is really how engaged and committed our researchers are and really thinking about the ways that we can make an impact in our communities. So this year we're trying something a bit different. We're having lectures in on our different campuses. We started out up on Burnaby Mountain, and now here we are in Vancouver. And I think just trying to, again, connect our researchers with our communities. So I'm really very, very excited, as I said, to have Carolyn's research here downtown with you this evening. And as I said, this is really thinking about highlighting the ways in which our researchers are making a difference to British Columbia. And this is a thematic area for Simon Fraser University. We are a public institution. We have obligations to make a difference for our communities. And I think this research really will highlight the ways in which we're doing that. So before we get started, I've got a little bit of housekeeping. And again, thanks to the technical team for AV support. And so for those of you on Zoom this evening, I want to just tell you that there is a closed captioning option, and there's a button at the bottom of your screen that you can hit. I would encourage you, if you have any problem, to send a message to hosts and panelists, and someone will respond to you with some help. So for the QA tonight, and this is when you can get your telephone out, we're going to be taking questions online. And also in person. Online, we're going to be using Slido, some interesting technology. It's a site you can access on your phone. I think you could just scan that QR code for those of you who'd like to do it that way. You can post your questions online, or we'll also take questions in the room this evening as well. So for this to work, you go to Slido on your web browser. There's a code there that you can see, SFU PFL, and then you can submit your questions online, and I'll be scanning those during the Q&A period. We do have community guidelines in terms of the way we engage, and I think it's pretty obvious to everyone, but we expect people to be respectful in their dialogue with one another. I also just want to note that this evening's lecture is being recorded and will be available on the SFU Public Square website and YouTube channel. So now it is my great pleasure to move on to the main event and to introduce our speaker this evening, Colijn. Colijn it's going to take a while, Colijn, so you can sit down. I've got a lot to say about Colijn. She's so fantastic. Colijn is a Canada 150 chair in mathematics for evolution, infection, and public health, and a professor of statistics here at SFU. Her work focuses on the topics of mathematics, epidemiology, and the evolution of pathogens. Before joining Simon Fraser University, she earned a Bachelor of Science at UBC and then went on to York University and University of Waterloo for a master's and her PhD degrees. Her postdoctoral training took place with Michael McKay at McGill University and with Megan Murray studying epidemiology at Harvard. Since then, Colijn was awarded a Genome British Columbia grant to help map the spread of COVID-19 in British Columbia, where she led a team of researchers measuring how effective BC's health policies are in lowering risk of COVID-19. I think we're going to all learn a lot about what happened behind the scenes with modeling and with the advice to public health. So please join me in welcoming Colijn for the President's Faculty Lecture.
All right. Hi, everyone. Thank you so much for coming. And thank you so much to Joy Johnson's office and Joy for inviting me to be here tonight. It's great. And I'm really glad you mentioned that about, being a public institution and having a duty to engage with the public. I think that informed some of the work that we did in the pandemic because there was so much interest from the public. And so we felt as publicly funded scientists that it was part of our role to get into that work, to advise government when asked, and also to convey what we were thinking about and the dynamics that were going on and the analyses we were doing to the public. So I want to take you back. Sorry, I pressed the wrong button. I'm going to press it this way. I want to take you back to the very start of the pandemic. And I'm going to tell the stories about what data can be used to answer which kinds of questions and how data and theory played a role throughout the pandemic. And I'm going to start by taking you back to the very beginning. When there were the first signs of this pandemic, the COVID-19 pandemic, early in 2020. Some of the very first signs were unusual pneumonias with no identified cause. So there were 41 confirmed cases, all had pneumonias with unusual things. So fevers, coughs, muscle pains and aches or fatigue, and sputum production. These were quite severe cases. Some of them had the complications that are listed there. And when there's no causative agent, no virus or bacteria that they can say, oh, that's a pneumococcus or that's an influenza, then of course that's worrying, right? Because 41 people are super sick for an unknown reason. And that was really early in 2020. And that's kind of the first data stream you might get about a brand new pathogen that you're not monitoring and you don't know about yet, is really people get sick and you don't know why. So then you start looking, well, why? And you start looking for pathogens. And it was actually very quick and quite rapid that this pathogen, COVID-19 or SARS-CoV-2, was sequenced and the sequence was shared. And that led to the development of early diagnostic tests, including development of tests here in BC at BC CDC. Okay, you did it wrong again. Good, I'm gonna do it right. You can see some of the news, some of this, sorry if this takes you back to a terrible and dark time, which it was a dark time, but news reports started coming out immediately about numbers of deaths, tech stock, biotech stocks soaring, other stocks tumbling, timelines, images there early in 2020 of many people masking in regions where masking is more normalized and people remembered still what SARS-1 brought and how scary SARS-1 was. So these things ramped up pretty quickly. And by January 10th, there were already confirmed cases outside of China. January 10th, if you can't see, is before the start of this graph, but it goes up hundreds and then 4,500 is at the top. So by kind of mid-February or early February, I guess, there were on the order of hundreds of confirmed cases outside of China. And this paper by Wu et al. In Lancet was one of the earliest estimates of a mathematical parameter. I think it was an R0 estimate that came from looking at people who had flights that had confirmed cases and then how many people had been infected onwards outside of the region. That's in China, obviously, but then outside of that too. So you can see the number of confirmed cases growing in that kind of exponential shape. And that was early on. In early February, we started talking in my group about these data. Papers were already coming out. I forget if I have a slide showing those papers. No, I don't. I'm going to go back. papers were already coming out with international data and with modeling estimates. And we thought, and there wasn't data here from BC, that this virus had not come here yet. But we wanted to get involved. We were infectious disease modelers and data people. And so we had a hackathon at SFU in the big data hub. And we had 6 different teams, one looking at flight data, one looking at severity data, and looking at case fatality rates and estimation, and various other teams. And that was really right. We started planning that in early February. I think it was February 18th. So right when there were just a few hundred cases, there was already literature coming out. There were almost as many papers coming out some days as there were new cases reported. It was a really rapid response from the scientific community. Okay, so I want to focus in this first part about really the early questions you want to know in a pandemic, you want to know the answers to, and how you would get data to estimate those things and answer those questions. So the first thing you might want to know, so new cases of a brand new pathogen are likely to start arriving in your community, and you don't really know what to look for. You might look for these unexplained pneumonias, but already at that time, I think it was established that not all infections were going to be severe pneumonias. If you wait for people to have severe pneumonia before you know what's going on, it's going to be too slow. So you want to think about actually infections, not just people with, terrible though it is, you don't only think about the people who have really severe disease. So you might want to know, how fast do I expect this outbreak to grow? You might want to know how soon do I need to find someone who's infected in order to prevent them infecting others? Do I need to get them into quarantine 3 minutes after they're infected? Probably not. 3 days after they're infected? Yeah, maybe. And the answers to those questions depend, and they're really hard to measure. So I want to talk briefly about the serial interval, the incubation period, and transmission before symptoms. And this was the topic of a paper that we wrote in the 2 weeks after that hackathon. So in the couple of weeks during February 2020. So I'll just give you some concepts. So we have 2 people, A and B. They're very creatively named, A and B. And so time is going along from left to right. And at some time with this big arrow here, A infects B. Okay, and at some later time after A gets infected, it takes a little while before the immune system is kicked in and there's enough virus and it's attacking and there's inflammation, and A gets symptoms. So there's little star points here when A and B are getting their symptoms, okay? So A infects B, A gets symptoms, and later B has symptoms. Okay, so we call the serial interval the time between when A has symptoms and when B has symptoms. And that's something you can observe, right? You can watch, okay, when did you start feeling sick? Tuesday? How about you? When did you start feeling sick? Sunday. Okay, that's 5 days. And to other people, it might be 1 day or 2 days, right? So you can collect all these. The incubation period is different. That's the time between when you get infected and when you get symptoms. So incubation period is the time between getting infected and getting symptoms. Serial interval is the time between symptoms in A and symptoms in the unfortunate person that A infected. Okay, got it? Right, so you see on this picture, there's a gap here, right? Which is this incubation period here. This gray line is actually longer than the red line. I hope you can see that. Oh, you can't see the pointer. I don't have a pointer. All right, gray line at the top, where it says incubation period, is longer than the red line at the bottom, where it says serial interval. And the difference between those 2 lines is the amount of time before A got symptoms that A infected B, right? And that's actually really important, because if you only know someone's sick when they have symptoms, and that's when you stop them, that's when you say, oh, you got to quarantine if you have symptoms, but they've already infected their contacts, you're too late. And that's what was happening with this virus, with SARS-CoV-2 and with COVID-19. And that was not happening with SARS-1. With SARS-1, transmission was after symptoms. And that really is one of the key things still, throughout this whole time that made SARS-2 so different than SARS-1. Okay, so back to the stories of data. What data would you need to understand these intervals, the gray line, the red line, the short, the time before this transmission before symptoms? All right, well, for incubation period, what data you need? Well, you need to know when someone was infected, right? And then you need to know when they got symptoms. And you need to know that not just for one person, you need lots of them so that you can do a statistical estimate and understand how variable that's likely to be and what the mean is. So, okay, that's good. That's nice, clear, except for one major problem, which is it's hard to know exactly when someone was infected, right? Infection is a hidden event. We don't see it. We don't get to see the viruses going along through the air or the droplets and getting in there. So we have to kind of make guesses about, based on epidemiology and like boots on the ground research. For serial intervals, you need even more. You need one more piece, right? You need who infected whom. You need the relationship between A and B. Otherwise, you can't know the time intervals of symptoms between them. Okay, so this all means careful tracking of exposures and contacts, right? It's personal data. Where were people? When did they get exposed? Who's married to whom? Who's in a household with whom? That's very detailed personal data. It was very generously shared very early on by a group in Singapore, and also completely separately by, there was a shopping mall outbreak in Tianjin, China, and our colleague translated the whole little map that they had made. And those data are actually still being used. It's so rare to get these data publicly that these papers, ours has been cited many hundreds of times. There's another one with the same data sets similarly, and these estimates are basically still used because it's very hard to share and obtain these data. But they're critically important, right? Because you need to know whether transmission's happening before symptoms. You need to know how fast you expect cases to grow. You need to know the timing of interventions. How much is contact tracing going to help, right? So what did we do? We thought, well, you can know which mall people went to, and you can know whether they went to so-and-so church, and you can know what you think their exposure might be, but you don't really know who infected whom. There might be missing people. There might be many exposures that could have infected someone, and you don't know which one it was. So we developed new mathematical modeling that allowed us to estimate how much transmission happens before symptoms by estimating these two numbers, this incubation period and serial interval. And we were able to account for missing people, the possibility of missing people, and the possibility of multiple exposures. So that was nice. That was right after this hackathon. And what we learned is that about 40% of the transmissions in these data sets were happening before symptoms. So that was already very different from SARS-1 and really presents problems for control measures that are based on noticing symptoms and then doing something. Because if you notice symptoms and then you test and then you do something, you may have missed already 40 to 60% of the transmission. We also learned that transmission is faster, about 4-ish days for the serial interval than it was for SARS-1. So modeling estimates were using these longer numbers from SARS-1, 7 to 9 days, and actually it looks like it was quite a bit shorter. Okay, so that was really early on. These are key parameters that all the modeling and all the edifice is built on after that. I want to take a step back, and thanks to ChatGPT with DALL-E for the cool image of conceptual stepping back on the bottom there, and think about, not just this early point, but then throughout the arc of the pandemic, what were some of the questions? What are some of the data streams and data sets that can be used? And what's some of the new theory that I think can help us set the stage as we risk the next pandemics? Sorry, again, not with the good news. Okay, so I've put, the Y got a little trailing on the bottom there. I don't know how that happened. Oh, there it is. On this one, it's different. That's great. This one, the bottom line has theor, and then the Y is like sticking down under the box, which is great. Okay, so it's good here. So I'm going to I've talked about sort of type 1. I have 4 data types here. Really individual data, these about risk and timing, these things I've just said about who's exposed and who infected whom, and the details and the time, and the red line, the green line, or the gray line. The second type I will talk about is data and theory used to make projections, and probably some of you will remember these projections. Questions like how fast we'll see things, how many cases, when and where, the healthcare burden, how many others does someone infect that are not. I won't talk as much, just a little bit about number 3, data around interventions that might be vaccine efficacy, impacts of different types of vaccine rollouts, which we also worked on. I won't talk about it as much. And then the last one I will get into at the end, data on evolution and the modeling on the evolution of the virus, and analysis of sequence data that shed light on where the virus comes from, but also how it's spreading, how it's moving around the world, and among people. Okay, so that's kind of a big picture, 4 broad data types and 4 broad classes of questions that you can answer with those data types. All right, so I'm going to move into type 2 projections. And this is some of the work we did early on the pandemic, kind of next after this estimates of these intervals that I just described. So you probably, some of you who are based in BC at the time probably remember these case curves. The blue dots are daily reported cases from positive PCR tests, and you have time going along on the bottom. And you pull together, information about timing, testing policies, information about non-pharmaceutical interventions. You remember the restaurants were closed and you were or weren't allowed to go for a walk with your friends or have your friends over, or your family. And if you were single, you had separate, all of these things that we probably many people hope to forget, but you do remember them. So we pulled together information about that and, are asked what's likely to happen next, and are asked to estimate, what might happen if we relaxed some measure, or, what might happen if we introduced some other measure. So we want to there's a question mark on the right-hand side there. What's going to happen? Well, I don't know. It's a big, noisy, messy set of data. So I'll tell you a little bit about one of the models that we used. And we use models, really mathematical models, because, well, they're useful to answer this question. And that's because they're able to synthesize a lot of different kinds of data. And we don't often think of models as a form of evidence synthesis, but I think they are. They can bring together information from these contact lists and so on that I just described about the pace. They can pull in information about severity, about age. They can bring in information about the time it takes between someone getting tested and having an episode or needing acute care, so healthcare burden questions. They can bring in information about behavior, just the fraction of the people who are engaging in distancing measures. There was a survey about this at the time that informed our model. And they can also bring in information about mechanisms, right? We know how infectious diseases work among humans. Humans get them from each other. So COVID doesn't rain on you from the sky like rain does. You need to be in contact with someone else. And that's fundamentally a mechanism where the amount of contact in the population drives the amount of infectious disease, especially early on when no one's immune yet. Okay, so I've got my little cartoon. I love cartoons as a way of explaining ideas and models. The little houses are evident. The green people are people who are engaging in distancing. They're able to stay home. The pink people are people who have a job or otherwise need to leave the house and have to have contact. So when they leave the house, they're more likely to bump into other pink people who are also out of the house. And so that gives you some dynamics in the model that you can encode with distancers and non-distancers, and then use that to estimate what's the impact of distancing. So we develop the math, we fit the math to the data, and then we forecast and estimate the impacts of distancing measures. So that was a whole phase of the pandemic for us. And it was used in BC. It was a great collaboration with the BC CDC. We had a group from SFU, UBC, some fantastic statistical ecologists from Fisheries and Oceans, someone from Vancouver from the Children's Hospital. And we developed this model, and this shows kind of the estimates of what would happen if the contact rates were changed, and they had them in the pink plots, and you can see Dr. Henry in an earlier time before this data, but earlier time kind of explaining those dynamics in one of the public health briefings. Okay. I'm going to move on now to, I didn't do another stepping back slide, and stepping back, you can imagine you stepped back and now you're going to step into a different part of the pandemic and also a different type of data. And this is more the type of data that's the bread and butter of our research group at SFU. And that is sequencing data. So people get infected, they get tested, and then sometimes, and then throughout the pandemic, most of the time in BC, that virus is actually sequenced. So we read the RNA of the virus and really could detect even very small amounts of variation between people's viruses. If that's all done systematically and shared, especially with extra information linked to other information, it allows us to do a lot of interesting things and really get a high resolution picture of how our infections are evolving and spreading. So you've probably recognized some of these. We can track the evolution of the virus and see what new variants are popping into our population, or even new variants that are emerging here. That didn't happen. Substantial new ones didn't emerge in BC, but they emerged somewhere. We can look at the mutations. We can detect new variants of concern soon after they emerge. We can look at how fast variants are growing and say, oh, look, we could see Omicron coming in November of 2021 because there was sequencing, global sequencing efforts let us see how fast it was rising and how it had risen across the world. The same with Alpha, the same with Delta. It allows us to connect mutations to severity, transmissibility, and immune evasion, which are the kind of triple whammy that you want to worry about. If you have a transmissible variant that's spreading among immunized people and causing severe disease, that would be really scary. So it allows that. And it allows us to understand how the virus is moving from person to person and also around the world. So those are the last two are the things I'm going to talk about, because those are some of the areas that we work in the group. Good, I'm just checking that the weird formatting on this computer is not on there, which it's not. So that's great. So I'll talk briefly about where was the virus in the past from the point of view of a person to person. So let's say I have all these sequences. And actually here, the work that we've done was more in bacteria than in viruses, but you can just suspend your disbelief for a moment. The idea is we track this small variation in the pathogen, and we use the genetic information to learn about who infected whom. We also want to know, we want to know who infected whom, but we also want to know how much confidence we have in that. Do we think A infected B, like maybe, or do we think A infected B 95%, that was what happened? We really need to protect people like B from being exposed in the kinds of ways that happened when A infected B. Okay, we want to know, how long between infections, and we want to know how many infections we're missing and where are we missing them. So sequences, the RNA or DNA of our pathogens, offer this potential because they offer this high resolution view of, which viruses or bacteria are related to which ones. But that's not perfect. Those sequences don't leap out of the sequencing machine and say, hey, I guess, it was Bob and Alice. Yeah, okay, that's where it was. You need to actually do analyses and quantify that information to pull out this kind of picture. The picture here shows possible stories of how these cartoon people might have infected each other. And then the thickness of the lines denotes how confident we are about those inferences. Okay, so I'll briefly describe the how. We use this thing called a phylogenetic tree, and it's kind of like a tree of life. You've probably seen the trees of life where you have the humans and the chimpanzees and the gorillas, and it shows the patterns of relatedness and who's related to whom, just like that, but with your viruses or your bacteria. And the core idea, of course, is that if two people's viruses are really, really closely related, they're much more likely to have infected each other than if they're really distant. Right? Because it takes time for viruses to acquire genetic differences. And so if lots and lots of time has elapsed, it's more likely there's lots and lots of extra transmission events in between there and they didn't infect each other. Okay, but it's a challenge because, like I said, the data don't jump off the page and tell the story. You actually need to do modeling and inference to pull those stories out of these data. And it's quite complex. The data sets are quite complex. Okay, so I'll just briefly say how we do it. We color this tree in. We have this tree of life. We color it in. And we have some rules that we use to color it in. So everybody gets a color. It's not necessarily a real color. It could be a number, but for visualization, we like to have a color. Not everyone has to be sampled. The virus only gets to be in one person at a time. Each individual, the dividing line of viruses are only in, each individual virus is only in one person at a time. And the colors have to stay connected. Okay, so we can set up that rule in our computer and we can generate a possible coloring. And then we can say, that one's really unlikely. That one meant that, somebody infected somebody else with flu 6 months later, but people recover from flu in 3 days to a week. You're not gonna have flu today and then next March, you're infecting your friend with flu. No. So we can make these colorings and we can say some of them are really likely and some of them are really unlikely. And then we can sample those. And there's a statistical technique for that called MCMC. And the result is that you get a whole big collection of possible who infected whom stories and who infected whom and when with timing information. And from that, you can pull out useful things. Some idea of who infected whom and when and how uncertain it is, how long it took, the placement of missing cases. It does leave a lot of uncertainty. It's not suitable for all infectious diseases. I would say it's more suitable for something like TB than for COVID, but these methods the tweaks of these methods are used for COVID too, to understand transmission using sequence data. There's some applications there listed. We looked at TB transmission in Norway. I say recent, even though it was 2013. That means recent for the people, not recent for the right now. It's of course 10 years later. It's also been used to look at TB transmission in the Republic of Moldova and in South Africa. It's been used to look at Ebola virus transmission and other pathogens. Okay, so last method, did want to include this. This is from the same paper from the Republic of Moldova, and it shows in the middle that tree of life. So the little pink dots are basically at the tips. And each of those concentric circles around there is an antibiotic. You can see the names of the antibiotics going down from the top of the circle towards the middle. And the colored dots show whether that individual's TB was resistant to that antibiotic. So you can see there's quite a lot of resistant TB circulating in that area. And so it's important to understand, how that resistance is transmitting and how that resistance is evolving and rising in that population, because you don't want drug-resistant tuberculosis. This is a bad idea. It's a very different pathogen than COVID, but still important to have the right methods to understand its transmission. Okay, final topic. We just talked a bit about where was the virus in the past among a group of people. Did Alice infect Bob, or did Bob infect Eve? We can also think about where the virus was in the past at a global scale. So again, we have our virus sequences as in a kind of tree of life. And that tree of life shows patterns of ancestry and descent in the virus. And we try to color those trees in according to where the virus was in the past. But of course, what can happen is some places sample more than other places, and that affects the quality of our estimates. And in fact, even in COVID, early on, there was Canadian data thinking, kind of imagining that our COVID was coming from the EU. I think it was probably coming from the US, but the US wasn't doing any sequencing. So there weren't any US samples in these trees. And when you color the tree in, you get apparent European ancestry was probably coming in through the land border at the time, and we just didn't have the sequences to know that. So together with a student who's actually here, Yexuan Song, we did some work developing a new method to account for that difference and try to get better inferences of where viruses were in the past. So how these methods work, there's the tree, and here we only have 2 locations, and we want to know where the virus was at those internal nodes in the past. You can imagine time is running down the page from the top to the bottom. The standard method uses a mathematical model, and it describes changes in location as if location is kind of evolving along the tree. So the location of the virus changes from blue to yellow or yellow to blue as the virus moves around. What we did, and I don't know why it's hanging off the bottom there, but we added something to that mathematical model that said, okay, well, you're less likely to see nodes where you're not sampling. So if there were things from the US, they might well be in your tree, but you wouldn't have seen them. And if you add that idea into the model, you can actually get different results. This is data from an Ebola virus outbreak, the big one in 2014, 2015 in West Africa. And you can see that in the full data set in the paper that was published, there are red tips showing that, or there's kind of a red part of the tree showing the virus actually moved through Liberia. It went from Sierra Leone to Guinea through Liberia. But if you start messing with the sampling and you don't get all the data that they first published, you can actually completely miss that it went through Liberia at all. So it really affects how we understand how viruses move. So what Yexuan did and our team did was actually adjust for that. And this is simulated data again, and I've shown the arrows highlighting where by doing that, you can actually reconstruct the true location of the virus in a better way than with the standard method, right? So by those arrows, our new method gets the correct red dots by the arrows, and the standard method only gets the blue dots. And right now, that's kind of a proof of principle, but we have a collaboration with the Public Health Agency of Canada who want to use this to do better understanding of where COVID-19 was throughout Canada, to look at how it moved across provinces and how travel measures might have affected it coming into the country. Okay, so I'm going to step back again out of kind of methods entirely, and I'm going to just recap a bit, what data sources we have and why we have them, why we want them, and when we might want them in a new pandemic. So we have individuals and their contacts, and we might want to know these timing questions, right? Serial intervals, transmission before symptoms. And for that, we want that as soon as possible. As we move on, we might have, unfortunately, transmission locally, and we might have these aggregate case counts, hospitalizations, and we might want to be thinking, okay, what's it going to be? What's the burden going to look like? What happens if we closed restaurants? We then have immunization, vaccination, I won't talk about. We have behavioral data. I didn't talk about that too much, but there was a lot of interesting contact tracing apps and contact data and mobility data. Google's mobility streams, Facebook's data for good, TELUS had data. We looked at all of those things. And that behavioral stuff, vaccine attitudes, attitudes to infectious disease risk, those are all, important data streams. And then of course, sequencing, which I talked about extensively. So there's really a lot of different data streams and a lot of applications and questions, and you can match them up, which one goes with which. Okay, I want to say it's not just data, right? Data doesn't tell a story by itself. You actually need statistics, you need mathematical modeling, and you need links among the data. We actually wrote a paper to say this, which was felt a bit silly because it said essentially, hey, if you want to know whether something really dangerous is transmitting among immunized people, you need to know how dangerous it is, whether it's transmitting, and whether the people are immunized. But amazingly, those data are not often linked. The sequence data to the immune status, to the epidemiology, where were people exposed, and to the clinical outcomes, because outcomes happen later and it's a different data frame and it's not linked and you can't share it and there's ethics and But on the other hand, you do need to know, is something scary happening now, or is, and when would we know, right? Okay, there's another pitch for why mathematics and models. Models really bring together these different data sources from different domains that can be measured in very different ways, from lab assays to counting cases in the population. And they create a synthesis that's an interpretable picture that people can understand. All right, so data and models are great. I want to make one kind of cautionary point, which is a completely different point. There's a WHO guideline on ethical issues in public health surveillance. So it's easy, I recognize it's very easy for me as a modeler and a researcher to say, give me the data. It's great. Like, come on, give me the data. But of course, data doesn't rain down from us, right? Someone has to do work, and that work is done in a population that is essentially under surveillance, right? If you get tested and your data gets included in some public, government-owned repository or data frame, that impacts you and your community. So that raises ethical questions around the potential for misuse of data by government, around the potential for stigma, as happened extensively in the early days of, continued for 2 decades, I guess, of HIV, and maybe still in some places, and HCV and other infections can be stigmatized. So there are potential harms. This document, I think it's great because it outlines some of these ethical considerations, and it concludes actually that societies have an obligation to establish public health surveillance, to share aggregate data with populations and with the communities that are being affected, to engage with those communities, and with national and international public health. And in emergencies, they have an obligation to do it quickly in a timely way. That also points out that there are harms from not having that surveillance, from not having that robust, shared, timely information about infectious disease. You can't point to inequity and address inequity if you can't see it. You can't have people take power and be empowered over their own infectious disease risk if they can't see it. And it makes other comments. So I wanted to make that remark about, it's not just data. Data doesn't just come from the internet. We design what data are gathered, about whom they're gathered, with whom they're shared. And I think, of course, the pandemic required new data, and we developed extensive capacity to get this genomic surveillance data, especially, but in infectious disease surveillance. We developed new estimation and new modeling tools, and that was demanded by the pandemic, but also by the questions policymakers were asking of scientists throughout. I think we should, with appropriate ethics and governance, we should gather, analyze, link, and share infectious disease data. And I think we owe it to society to make the most of these data and get in front of what we would see in a new pandemic and make the most of them in terms of interventions and preventing and preparing for the next pandemic. Okay, I want to thank you all for coming, and to thank many colleagues, students and postdocs, some of whom are here, who worked with the team on this, and colleagues at the Public Health Agency and the BCCDC especially. So thank you.
Well, thank you so very much. That was fantastic. I've got a lot of questions. I get to ask a few questions first, but I'll get you to start thinking about your questions as well. So please join me, and we'll get going. And look at, there's already questions on Slido as well. So, you know, what year did you come to Simon Fraser University?
2018.
2018. So here you are, 2020, and all of a sudden this starts to rain down.
Yeah, 18 months later.
Unbelievable. I mean, we're so lucky that we were able to recruit you, I've got to say. But what was that like? Like, this is like your whole life prepared to be doing this kind of thing, and it's happening. What was that like for
Yeah,
you?
that's a great question. I wasn't actually working really in pandemic preparedness. I was working in infectious disease and in genomics and evolution. But of course, I was an infectious disease modeler, and there are not that many in this part of Canada. There are, of course, some. I think we did kind of know in infectious disease that a pandemic could come, right? We'd seen in the last 2 decades, SARS-1, MERS, Ebola virus, Zika virus, H1N1, you know, and that's just to name the ones at the top of my head. But yeah, so I think we knew a pandemic would come. I think it definitely felt early on that something big was coming, and that it was going to be important to understand what the data was and what we could do.
Yeah. And was that before the hackathon, or like, I'm just curious about that, like when you really thought this is going to be big?
Yeah, I would say before that, and that's one reason we did it.
Yeah.
You know, it was in the news, and I was thinking, is this still going to be in the news in 3 weeks? And it actually, yeah, this is not going anywhere. You know, when you saw those cases rising, curves. Yeah.
And so you, during the pandemic, you did engage with the media a lot.
Yeah.
It must have been challenging to, I mean, I felt like I was hanging on by my fingernails listening to the modeling. It must have been hard to explain this. Like, what were some of the techniques you used to help explain all of this to the public?
I think it was a learning curve for sure. One thing I remember was hearing an interview with Colijn from the National Committee on Immunization. And they were talking about new vaccines and developing mRNA vaccines in Canada. And she was talking about it. She's an expert in the area. And then they asked her some question about what the facility would have to be like, and what the timelines were like, and really what the logistics were like about producing a new mRNA vaccine in Canada. And she just said, oh, well, for that, you'd have to ask someone who knows anything about the logistics of this. And I thought that was fantastic, because it's so okay to say, that's really not my area. Yeah. So I think one trick is recognizing when they actually want to know something about, you know, okay, not just what the hospitalization numbers might be, but what that will mean for a reader if they turn up at that hospital with a broken leg. And of course, I don't know. But we also did get some metaphorical thinking going, and we got some really clear examples. I don't know if we have time to give you one or two of them.
Like, because metaphors can really help. Right?
Metaphors can really help. Okay, so waves. We've heard about pandemic waves, and I think now they are more like waves, but throughout all of 2020 and 2021, there were not really waves, you know, in the sense that, okay, imagine you're lying on the beach, right? And you've got your towel, and there's a wave. What does not happen is the wave only recedes because you jump off your towel and push it back. No, this is not what a wave does. A wave comes and then it goes. But what was happening in 2020 was more like sparks landing on dry grass, right? And gets put out because you put water on it, not because you sit back and you wait for the wave to recede and do nothing. So I think that was that kind of analogy around, you know, here's another one, also could be done with the fire, but I'll use the mice. So let's imagine you have 2 decisions you're going to make, right? You have decision 1 and decision 2. And you might be tempted to have there's some trade-offs between 1 and 2, and like they're kind of expensive. I kind of want to do number 2, but I also need a new roof. So, you know, I need to fix my stove, but I need a new roof. And what I'm going to do is, or let's say I need to call the pest control guy, but I also need a new roof. So what I'll do is I'll get the pest control guy to get rid of half the mice in the house. and then I'll still have money left over for the roof. No. If the mice are going to grow themselves, you don't want to get rid of half the mice. You got to seal the house and get rid of all the mice. Right. And that's sort of a metaphor for the closed borders, you know, and like what New Zealand was doing or what Atlantic Canada was doing. I think some of those were helpful. Another one that we just struggled with so much was just nonlinearity. And that what I just said was an example. You don't get halfway done by putting out half a fire or removing half the mice. That doesn't get you halfway to your outcome that you want of having no mice and no fire. You can't get half the fire. You can't put out half the fire. You got to get the whole fire. But another one around nonlinearity is around exponential growth, which you heard about. There were cartoons about it. Like there's everything about it, flattening the curve, the whole thing. But if you have 12 cases and it takes you 2 weeks to get to 50 cases, how many weeks is it going to take you to get to 200 cases?
Yeah.
Only 2 more,
Yeah.
not 6 more, not 8 more, not maybe it's slow, it's not exponential yet. No. So that was one that we struggled with and we tried. I think another thing that's important to do and is still a challenge is really communicating uncertainty.
Yeah.
So
Yeah.
you have with modeling, people either believe them and, oh, this means there's going to be 491.2 cases next week, or they don't believe them at all. Like they mean nothing. And it's something in between. I think that's a challenge.
Yeah, absolutely. I'm going to get to some of these questions on Slido, and I will also check in with the audience as well. So here's a question from Nat. I see a few variables. Okay, some of these are more technical than others, I've got to tell you. I see a few variables impacting transmission that could be tested in reductionist systems with good quantitative empirical data. Is there much of an effort there? It would complement the ecological modeling studies, it seems. Did you understand that question?
Yeah, so I think so. So there were a lot of factors that impacted transmission, and people did do statistical estimates to try to tease apart the impacts of different NPIs, different non-pharmaceutical interventions. So the masking and the plexiglass barriers and the hand washing and the hand sanitizer and the social bubbling and the keeping people out and the reducing. And one challenge with that is there's a lot of confounding there because a lot of jurisdictions rolled out a suite of measures at a time. But yeah, there were papers that did that, and they, as far as I remember, got mixed results. There was one that had Facebook data, which is a bit of a limitation. But it was one of the only ones that actually did harm was the plexiglass barriers. I remember that. And that's probably because they reduced airflow. And so but air carrying virus could hang around in the room more. But yeah, there were some efforts.
Interesting. Interesting. Here's another question from Linda, and I will turn to the audience, see if people want to ask a question as well, and I'll repeat it for those online. If a pandemic happens every 100 years, would the chance of a pandemic now be any different as population increases, encroach onto land inhabited by wildlife, and we travel everywhere?
Yeah, thank you. I think that's a great question. And actually, people have termed this era the pandemicine because there is a real concern about exactly that, about animal ranges changing, mosquitoes, ticks, bats, rats, about human density increasing, about climate change driving.
Give me some good news.
Yeah, right. Climate change driving changes in that, and indeed flight data, it makes it, human mobility patterns make it much more difficult to contain something, especially something that can transmit without symptoms. So I don't think our current pandemic makes the next one any less likely. I hope that it makes it more likely that we'll be prepared and we'll be able to prevent the next one.
No, absolutely. Yeah. I mean, it just is, I think it is really clear that there will be other pandemics, and
there've always been pandemics. There will always be pandemics. There'll always be
absolutely.
evolution. There'll always be gravity. Like these things are going to happen.
Yeah, absolutely. Just going to check in and see if anyone in the audience, yes, if you wouldn't mind just using your loud voice at the very back or toward the back there, and then I'll repeat the question. Yeah. Right. So there's great. Thank you. So there's 3 things there. Errors in modeling, modes of transmission, cardboard, other kind of things, whether there was ever any evidence. And the first one was around not being vaccinated and the impact on the modeling.
Yeah, so I think the role of unvaccinated people in transmission has, of course, really changed. Right now, almost everyone has had COVID. They would have had some immunity. The early vaccination course, protection against infection has probably waned. Protection against severe disease, maybe less so. So I think that answer really changed with the stage of the pandemic and with what was circulating. The cardboard, yeah, I mean, it is amazing to me. Like even at the time that, we did this analysis in summer of 2020, and maybe fall of 2020 about outbreaks and whether it would be better to do distancing or bubbling and under what circumstances. And there were these famous outbreaks where 80 people got infected in 2 hours. And the very idea that this was all going to be because 80 people managed to touch the same surface and put it in their eye and in the 2-hour period at a wedding or a choir concert seemed really unintuitive to me. I doubt there was ever a large risk from your groceries coming in, from droplets there. I think that these large, the majority of transmissions were direct person to person, not by a cardboard or a playground or anything like that. So that kind of amazed me that people, that was promoted so much, and that it lasted so long, really.
People
with the
were
hand
washing their
people were
groceries
wiping
and-
their groceries.
Yeah.
And even the hand washing is critical. It's great to wash your hands. Don't stop washing your hands. A really good idea. But I don't think you can prevent COVID by washing your hands. And that was so, maybe a little bit, but I think we do need to deal with the fact that air is a thing. And we don't say to people, if you're worried about gastrointestinal disease, we should boil the water at your house. Like, go boil your own water. We don't do that. We raised cities 1.5 feet. We delivered a public health water infrastructure. We got rid of major public health issues by cleaning water. And I think eventually it took time to do that. It took time to recognize that that was necessary. But I think we will start thinking of air more the same way. And I hope we do that before the next dire airborne thing comes out of a bat or a rat or a pangolin somewhere, in 2035 or in 2050.
Yeah. So
And then the last one, errors in modeling. Yes, models are wrong. All models are wrong. We cannot simulate the world in its full complexity. We hope that models are useful. Models cannot predict human behavior. So models often overshot in the pandemic. If you just take that exponential thing and you forecast it outward and you wait until everyone's immune and then it falls back again, you will get an overshoot compared to what we saw. And partly actually seeing model projections and how the level of interest from the public and the fact that models were used to help inform policymakers, I think that partly, models were in a way self-defeating. Like, you can say that this is another one of my favorite analogies, so I'm glad it came up. If you're using headlights and your headlights show you that there's a moose in the road and you veer and you don't hit the moose, you don't say, drat those headlights, their prediction was wrong. No. The headlights are a tool that help you see what's on the road ahead and help you make decisions. And sometimes models were like that. I think sometimes they were actually overestimates or sometimes underestimates for reasons that are hard to unpack and differed in different situations.
It's interesting though, because we saw the public taking up those models and treating them like fact.
Right.
And I think that's what really got complicated. And again, it's that uncertainty, right? Like, how do people really understand uncertainty?
And what is a model and what is it trying to answer? It's often not trying to predict the future. It's trying to show scenarios and trying to show, what is the path that you're on, not predict whether you're going to continue on it.
Yeah, so interesting. See, now I've got, yeah. Okay, so I'll go here. There's 3 questions. I'm going to go here, here, and here, and then I'll go back online. Yeah.
Thank you for sharing your knowledge and experience. My question is about the elderly. Not elderly people died or got infected. Were you surprised by it, or were you expecting some data from that? And then what was the contribution from South Africa or Africa in general? Because in terms of international data collection, South Africa was quite prominent.
Yeah, so I'm going to repeat the question for those online. The question really is about the elderly and the high death rate that we saw amongst the elderly. And so comment on that. And then also about South Africa and data that came from South Africa.
Yeah, so I guess I'm not a clinical person. And so I didn't really have an intuition going in about what I would expect in terms of differences in severity with age. I don't think it's terribly surprising that the elderly would be more at risk, but I think the fact that they were 100 times more at risk was surprising. It wasn't just that they were 2 or 3 times as much at risk. So I think that was surprising. And of course, it was a very important part of how the first stage of the pandemic was managed. South Africa does have a fantastic genomic surveillance program, and they were very generous in sharing their
Yeah.
data, especially at Omicron time. And it was not to the benefit of South Africa, because travel measures were laid out that were very punitive to South Africa at a time when actually the only real association with South Africa specifically was that they had shared their data.
Yeah.
And so I think that's really a lesson in understanding that what's seen in data is not what's in the world. It's not the only things in the world. And we do have to be, incentivizing and not disincentivizing public health data sharing. I think it's really important. By the time those measures were introduced against South Africa, the Omicron was already spreading in Norway. It was already at a Christmas party in Norway. It was probably all over Europe, and we only know it was at a Christmas party in Norway because Norway has great surveillance and data sharing.
Yeah, so interesting. You do not want to de-incentivize countries from sharing their
Yeah.
data, and those kind of stories did create a chill. There's no doubt
Absolutely.
about it.
Yep.
Yeah, it's interesting. Carol, I saw you had it.
So my question has to do with the fact that in Canada, we've got 10 different provinces administering health in 10 different ways. And that meant that there are different measures taken in each province. And thinking about the school kids, in some provinces, schools were kept open, in other provinces, they're open and closed and open and closed, and other provinces had different distancing measures in schools. Did that actually end up providing a helpful set of comparative data, given that there are all these different ways of handling the crisis? And although it was very inconsistent and annoying for people living in different provinces who couldn't keep track of the rules, especially if you're traveling, back and forth, but did it actually provide useful comparative data?
Yeah, so again, I'm going to repeat the question for those people online, and it is interesting. We had all these 10 provinces, different, and even within provinces, different approaches in terms of restrictions, schools open, closed, etc. And so did that provide us with some insights? And I imagine that's still getting unpicked to some degree.
Yeah, I think it probably is. I'm going to say I imagine it did. That's not an area that I worked in specifically, like taking that natural experiment and saying, okay, so what is the role of a school closure, given that all these different things happened and some people had schools open at some times. But I think that kind of thing does provide useful comparative data. I will say, I think it, in some sense, was a benefit for Canada to be able to have those nuanced approaches that were context specific. I think if the Yukon and the Atlantic provinces had to be under the same national rule as Toronto and Montreal, they would not have been able to meet their own needs. And they don't have the same hospital capacity as Toronto and Montreal. And so they couldn't have taken the same route. So I think in some sense it was an advantage, and also was, protective for some Canadians against what may have been suboptimal decisions, and no one can make perfect decisions throughout the whole time. So it does have its advantages, I guess, but it certainly has its frustrations when it comes to coordination and data sharing.
Yeah, it's interesting too, because we also had public health officers also having to make decisions about the spread, but also about protecting our hospital system, number one, and also about keeping our economy going. I mean, there were all these other factors, and about our young people and their isolation. It's like, it is complicated in terms of those decision making. I don't envy them, that's for sure.
Yeah.
I'm going to take that one more question here, and then I'm going to go back online. So please.
Dr. Carolyn, the transmission rates from SARS-2 down to SARS-1, I think you said went from 7 days to 3 days. So the next pandemic goes from 1 day to 2 days. Does that make your job a lot more difficult? Do we have to get faster methods from the policy holders of the government to make sure you guys get that data faster? Because I see a weakness there. I think the data comes to you faster.
Yeah, I think that's true. Well, of course. Oh, sorry. Repeat the question. Yeah. So I think the question is, if the pace of transmission can get faster, and then even faster, does that have implications for what data speed has to be, and whether data can get to analysts and scientists quickly enough? I don't think we need data, at the same exact time frame as transmission, but I do think faster transmission poses a huge challenge. Flu tends to have a serial interval of 1 to 3 days. So a flu pandemic, which is what people had thought of before SARS-CoV-2 as, in terms of pandemic preparedness, you would need to think about something that can spread in a couple of days and does have very fast dynamics. But with flus, most of the population has some partial immunity to flu. And so you wouldn't expect an entire population of like completely dry wild grasses, with the sparks, the way we had with SARS-CoV-2, because we were completely immunologically naive to it pretty much. I think we do need, and public societies should be demanding robust infectious disease surveillance. I think you and we have a right to know what infectious diseases are circulating, how they're evolving, and at what levels they're circulating, so that we can advocate for ourselves. People need to know if those are different in different communities. If there are some communities that are more at risk and that risk has health consequences, then they need to act and advocate for themselves. So I do think we need those data, and I think we need the infrastructure to be monitoring what viruses are out there, what they're doing, what impacts they're having, and to be ready so that we can be positioned as best possible to prevent further pandemics.
Thank you for that. I'm going to return to some of the questions, and I get a kick out of this one, and it's about another pernicious infectious issue, and that's about modeling around, is it possible to actually use modeling to look at the spread of false information and how that impacts the spread of virus? Like, is that something that one could also take a look at, in terms of your work?
Yeah, so people do use modeling to look at the spread of misinformation, and they look at, Twitter data when it was available for researchers to use. And they look at other online data, and indeed misinformation can spread like a virus. It doesn't always, it sort of has to land in fertile ground, and it has to be in a context. SFU does have a Digital Democracies Institute led by Wendy Chun, the other Canada 150 research chair at SFU. And I think they do look at some of those tools, and it's an absolutely fascinating area. I think it's getting worse. One of the things I wanted to bring up, in our discussion was, the need for continued public trust, and the role of universities, and the role of data, and the role of the scientific community in public trust. I think this is a dangerous time for journalism. We had so much high quality journalism in the pandemic. Journalists who, I had a journalist from the Vancouver Sun. I've momentarily dropped his name, but it'll probably come to me, Nathan. Anyway, apologies to Nathan if he's out there listening, but he actually downloaded our code from the GitHub for our paper on the vaccine rollout, and ran his own scenarios and wrote an article about it in the Vancouver Sun. And it was just fantastic to see that level of engagement and high quality journalism throughout from so many. And I do think we need to combat misinformation, and we need to be thinking about these things and how to support, science communication and journalism, and how as universities we can support public trust.
Yeah, no, it's so important, because even in the midst of the pandemic, with all that misinformation and this very, I would say, volatile, but also divided opinions about a lot of the measures, like, scientists were being threatened. Like, it was not fun. And I think we really have to applaud our scientists who leaned into this area, and went out and shared information. But I totally agree with you. It also, because there was so much misinformation, I think it was, in some ways, it was a great time for science, but also we also saw the other side about how quickly that could erode. It's so interesting.
Somewhat, and I think much more so in the last year or year and a half. But actually in BC, we had such a high vaccine uptake. People were so interested in vaccines. There was such clear, consistent communication, largely from public health and from science. I think we did a lot of these things incredibly well in Canada compared to in some parts of the US and some other areas. But I do think we need, especially as we continue to see journalism undermined, and social media and internet media, and people not paying for news, I think we do need to really keep on that and keep thinking about it. I'm delighted that we did have such a high level of public trust throughout, and I think it made BC a great place to be by and large. And there were points when that broke down in different ways, and when the scientific community and public health had, we had our disagreements throughout, but I think especially in those first 18 months, it was so high, and I think that was a credit to so many people and so many scientists and communicators and governments, and I hope that we can keep that up, but I think it is a real challenge.
No, I think it's so important. Absolutely. Here's a question, again, a technical question, but interesting. You have very interesting data sets to work with. What tools do you use in your team to visualize data that you're working with?
So, okay, well, I like ggplot. It's one of my favorites. They're very, very flexible, customizable ways to layer data and create effective visualizations.
And do you use it regularly for the modeling visualization? Of
Yeah,
data?
all the time.
Yeah.
Yep. And we like to work with evolutionary data, so phylogenetic trees, the kinds of tree of life that I showed in the
Yeah,
talk.
how things evolve over
How
time.
things evolve over time, coloring those in and making effective visualizations. And actually we have a researcher in the group who's one of his areas is how to visualize those kinds of objects, those evolutionary stories, while also trying to show some of the uncertainty in those relationships. So it is a nice, it's an interesting area because it's not always clear how to make an effective visualization of something where it's uncertain. You want to make a picture, but you don't want to make a picture that kind of too much, claims to have a truth that maybe you don't have. So yeah, we do use visualization tools quite a lot.
Yeah, interesting. It does help. I mean, to understand data in a different way. Yeah.
Yeah, and it helps with modeling, right? That's one of the roles of modeling. The reason I think modelers had so many questions from policymakers is that models can give a picture with time on the x-axis and scenarios on the y-axis showing what might happen and really interpreting lots of different data in one story.
Were you always interested in mathematics?
Was I always interested? Yeah, I think I started in physics. Physics was my scientific kind of first passion.
Yeah.
And then I got into more pure mathematics. And then I decided that I was quite interested in, things more in the world and in applications. So it was a bit of a route to
Yeah,
infectious
that's
disease.
always so
But yeah,
interesting. Yeah,
Yeah.
absolutely fascinating. So here's a question about immunity. You spoke of a time during the pandemic when nobody was immune yet. But nobody is totally immune now because current vaccines only reduce risk of acute illness, not totally of infection. I would agree with that. So what has modeling shown us about things like N95 masks and infection? I think the question is right now, what's the modeling saying about all of this?
right. So I haven't run estimates about effectiveness of masks. I did look into it. I think from what I've seen, N95s are going to do better than regular surgical masks. Estimates of community effects of masking in the community are all over the map.
Yeah.
And some studies had people, it's like, if the outcome is, did you get wet, but you only used your umbrella for 3 hours of your hike? Like, okay, yeah, you still got wet. Did the umbrella work? Well,
Yeah.
sure. But then you kept hiking for 6 hours. But, some of these estimates are all over the place. One of the studies I read that I think had 300 other studies in it came up with about an overall 20% reduction in the transmission rate, which could be huge. I mean, that could be it's not just 20% difference in cases. It could be actually a huge difference in cases. So I think masking is going to work. It's models can't really, what you can do with models is you can put an assumption about how much they work into the model and then see how it matches data. But if there's a lot of other things going on, there might be many ways to match the data. So models can't really estimate those impacts. You really need data about what happens to people prospectively when they do or don't do different things. And that's just really hard to come by.
Yeah. And are you modeling right now what's happening with what's happening with COVID?
Yes and no. We're still building methods around genomic data and how to leverage them and how to think about what they mean in terms of projections. One of the gaps I think is that we have this rich genomic information that tells us about the past, but then we have these really simple models that we use to try to understand the future. And of course, we will always have a better view of the past than the future. That's nothing new. But it would be great to use that rich data that we have from genomics to build better projections and better understanding of how variants compete with each other. So I'm working on those topics. I'm not doing kind of daily COVID projections or weekly projections of waves right now.
Okay.
To some extent we're still thinking about if a new wave comes, how can we know how big it's going to be?
Yeah.
So we're looking through data from all over the world to think about, are there systematic patterns we could use to pull out the risk in a population and how big a new wave might be.
Yeah, so interesting. Long COVID, is that something you're looking at? And the question really is about, there have been some estimates that 15-20% might end up with some kind of long COVID. Are you modeling it? You're looking at it?
Yeah, we are doing one project on that. I think 15 and 20% as a fraction of the number of infections is really, really high. I mean, if that would 15, 20% of that on every COVID infection, we'd all have long COVID by now. So I think that's high. I do think we have a serious issue, and it's one of the last sort of pieces I wanted to say about this kind of modeling before, moving back more into the genomics and evolution side, which is this, that let's imagine that the first time you get COVID, you don't get long COVID and everything's fine. Okay. So the next time you get COVID, does that actually give you information? Are you like just the kind of person who, for something about your own, your genetics or some factor, you're less at risk for long COVID than someone else? Or does that not give you much information? So I know a few people just anecdotally who got long COVID after their third COVID infection. I live in fear of this. I like being able to do my work and have my brain working. And like we all, this is something that terrifies me because I've had friends who are very profoundly affected by long COVID and lost, 2 years of their working life to it. So it's a huge problem when it happens. But the difference between those two things is actually really profound. So if I've had COVID twice, neither time did I have some long-term consequence that I'm yet aware of. So does that mean the third time I'm probably okay? Or does that say nothing about the third time? Or does that say something in between? And that's something we don't know. And if you model that difference, you find that if it's a random roulette roll of the roulette wheel every time, the burden of chronic disease in the population that we're going to see from getting 1 to 3 COVID infections a year for the next 10 years is really high. Like that could impact diabetes, cardiovascular disease, neurodegenerative disease, inflammatory disease. It can be a huge impact on the population. On the other hand, if there's some kind of concentrated risk, and the people who are at risk are, those ones at risk, and the others of us are mostly okay, it's incredibly inequitable for those individuals who are at risk, but the buildup of chronic disease in the population is much less. So that's the modeling we're doing. We can't really model, what is long COVID or the risk of it from an infection. You really need data on that, but this kind of population impact we can model.
So interesting. I love this question, and it's really about, if we could have done something differently. Now that you know what you know about the modeling, about transmission, are there certain things that would have been more effective, or certain actions that could have been taken?
Yeah, I mean, I will say recognizing the role of air earlier, and even now you see a lot more surface wiping and a lot less air cleaning than I think we will eventually settle on. I know that the word airborne is a scary word for the healthcare system, because if you say airborne, you trigger things that may be over the top for COVID. It may be that there's some continuum between completely only hand washing and wiping groceries and completely only like cigarette smoke in the air. I think there is a continuum of droplet sizes and transmission there. But I think we could have recognized the role of the air earlier. And I think we could still be doing more in terms of our infrastructure. And that's part of pandemic preparedness, right? Because the next thing, what if something had the fatality rate of MERS with 35% fatality rate and was airborne? That we would have to contain it. We couldn't do this thing of, oh, well, we're going to balance the factors and we're going to mitigate. We wouldn't do that. So having that infrastructure in place will protect us if that happens.
It's a huge investment. Obviously,
it's a huge investment.
I mean, I was, at SFU, we were, really having to invest, but also really think about it. So, yeah, I know.
And now
very important.
Now we're probably seeing the benefits of it.
Absolutely. Yeah. I also think, the other thing is, I think people's behavior has really changed.
Yeah.
I mean, I'm one of those people who used to go to work sick all the time thinking no big deal, but it really is not tolerated anymore. I think that human behavior has changed. In terms of when you have a cold or when you're not feeling well. I really do see it in my day-to-day life.
Yeah.
Interesting. It's interesting. The other thing I wanted to ask you about is about lessons for our public health leaders. And I mean, you were providing data, et cetera, but are there things based on we've talked about the importance of air, but are there other things that you would say to they were on the firing line, right? Poor Bonnie, every day.
Yeah, I cannot imagine how hard that that was and would have been. And yeah, I mean, I have so many things. I will not talk to you for the next hour here on this
Yeah.
one point, but I could. I would say, I think one thing I learned was how interested the public really was in the data and in the modeling and in the science. And, you know, the public are no dopes, like they're
Yeah.
smart and they can really process and understand a lot of nuance and a lot of if this, then that, and a lot of, you know, complexity. And I think there was such tremendous interest. So that really struck me. And I think the role of those clear, consistent communications was really strong. And I guess this gets us back to public trust. I think that continuing to have public trust by having a robust surveillance stream, by having those data available to communities, even where sometimes, you know, in the pandemic government was, you know, there's always a bit like, well, if those data get shared, they might get interpreted differently or misinterpreted. And we wouldn't want, you know, there can be a tendency, especially in an emergency, to just sort of claw back and be a bit more paternalistic. And I don't think the public needed that or wanted that. I think that, you know, if there was one thing I hope that we build together going forwards, it's great relations between the scientific community and public health around infectious disease. And that it's great data streams that can be shared, that can be ethically shared, that can be shared with the world, the way South Africa shared their data. I think we should all be doing that. And we should, where we can, be sharing those linkages too. So I think that public trust, the role of data, the role of the scientific community, I think we had a role to play in this high level of public trust that we had here in BC. And I hope that we can continue to have that. You know, it was all a bit ad hoc and built up in emergencies, but I think it should not be ad hoc. Now we know that this can happen. What the UK did with the COG consortium and the genomic data, they had, you know, everyone in the world benefited from those estimates. They also had all the COVID, so they had a lot of data to work with. You know, not all of it, but they had a lot of COVID. And I think they really did a service in sharing not just the data, the genomes, but the links and the analyses. So I hope that we can build that and build those relationships.
Well, my sense is that there's really been an awakening and understanding of the importance. We see that from the Public Health Agency of Canada. We do see that from, you know, BCCDC and from our public health officials as well. A real interest in trying to figure out how to do that. Yeah,
and the Canadian data situation, you know, with this federal, provincial, jurisdictional challenges, you know, one of the things that I thought was very surprising was that even in the Public Health Agency of Canada or the National Microbiology Lab, no one in Canada had access to all of Canada's SARS-CoV-2 genomes, certainly not with outcomes about severity. So no one would have been powered to detect something new coming out that happened across Ontario and Quebec, for example. No one in the whole country. And that's amazing. You know, so I hope that we will sort out those data sharing challenges and jurisdiction challenges in Canada.
The joys of being part of a federation, you know, like, so, it
Amazing.
it is challenging. I'm going to check in with the audience, see if there's anybody who wants to ask a question. Yes, please.
Okay, there's been a lot of talk of models. I don't expect you to get technical, but in the research on the COVID-19, was it all standard models from the epidemiology literature, or were new formulations developed? And related to that, of course, the models have to be statistically tested. So you had something in one of the slides that looked like some kind of linear expression. Were very standard multiple regression models used, or a variety of other methods, statistical methods?
Yeah, great question. So I won't try to repeat all of it, but the question is about what kinds of methods were used. Were new models developed? Were standard models rolled out? Were regression models? You know, what kinds of modeling was done? I hope I've captured the essence of the question. Honestly, we used all the things. We developed brand new estimation methods that used, for example, cluster size data to estimate transmission rates and using schools. We rolled out variations of the absolutely bog standard, susceptible, infectious, recovered. We tweaked those things in novel ways to capture the impact of distancing measures. We did use regression models. We didn't do that as much, but the community absolutely did that. We developed new ways to use genomes to estimate serial intervals, capturing different uncertainties. All of those things happened all the time throughout the whole pandemic. It was a vast effort. There were the contact data models, the behavioral models, there were new models that had cell phone data, there were models to use data from the contact tracing apps in the UK. Honestly, all of it, people did all the modeling of all the kinds.
It's so interesting. So did the quote science of modeling take a few steps forward as well? Because of this?
Absolutely. Yeah, for sure.
And were the modelers, I know there were a number of people doing the modeling. Were you in touch with each other also around some of this? Like, I'm just kind of curious.
Yeah. Oddly, our collaborations, even though everything was on Zoom and Teams, our collaborations became very, very local. So within BC, we had the BC COVID modeling group. We also had the internal collaboration with BC CDC, and that was such a focus. And then we had the national work, so very, very Canadian. And I co-direct a network of infectious disease modelers called CanMod, which has 42, I think, co-applicants, and there are 4 other networks, and there's a lot of overlap. So within Canada, we were very tightly connected. I think in terms of international links, a lot of us more kind of kept in touch with people in our international networks, but were much less close to their experiences. So it became sort of hyper local in a way that was somewhat unnecessary because everything was online. But I think modeling it's a little bit more like fire stations than space stations. You need one space station can do all the things, but you wouldn't want one fire station in Toronto to try to be coordinating, putting out all the fires. You really have your local context and your local questions. And so modeling groups became very hyper local with, Amy Herford and the Atlantic provinces really looking at, under the situation where you don't have any cases, how do you estimate RT? Well, you can't. You estimate what would happen if they landed, and what data do you use to do that compared to what was happening in Montreal was completely different.
And were there situations where the modelers disagreed because different models were being used, different types of data were being used, that there was that kind of?
Definitely. I think some of the major questions, there was such strong agreement though, like the question of, is the alpha variant going to rise here and when, and at what rate is it going to rise? Like any sensible model would probably have given you the same answers to those questions. I think one of the things that was great about the collaborative groups that I was in, the BC COVID-19 modeling group especially, but also the other groups, was that we could really hash out these differences and get to a consensus before putting out anything. And so I think that those disagreements, at least that I was aware of, kind of played out as we were developing the work rather than afterwards.
Yeah, so interesting.
But I guess one point where people disagreed was the vaccine rollout, whether it was a good idea to defer the second dose and whether it was a good idea to do a strict age-based rollout versus an age-based rollout that also factored in high contact or high risk workers. So those were some points. There were subtle points where people disagreed.
Yeah, that was so interesting.
Yeah.
We're winding down time-wise. I just want to see if there's any other points you wanted to kind of touch on this evening.
No,
I mean, it's really been interesting. There's lots more questions here, and I know the audience is just, it's really been fascinating.
It's been great. No, thank you so much for everyone's questions. It's always great to have such a high level of engagement for a talk. So it's fantastic.
Well, I've learned some things this evening, and just so proud of the work that you've done.
Thank
and
you.
The group. Great to have your group here as well, and really lovely to hear from you. So please join me in thanking.
Thank you.
Carolyn. So I do have just a few final little housekeeping bits and pieces. I've got to get my notes out here. So thanks. And thanks to everyone for coming this evening. I want to let you know just a few more thank yous. I want to thank the technical team for helping us broadcast the event from the Fletcher Challenge Theatre. I want to thank the Public Square team again, Janet, Seth, Gabby, Savannah, Takes a Village, Nola, Gabriel, Sophia, and Ina. And I want to thank our volunteers. We have volunteers who've helped us this evening as well. I hope you enjoyed tonight's event, and we have another President's Faculty Lecture coming up in the new year on January 23rd. And it's again, I just want to remind, we're focusing on challenges in British Columbia. We talked about COVID. Before that, we talked about agri-tech with Woo-Soo Kim. And our next one is titled A Sustainable Housing Future. And I think we can all agree that housing is also another challenge for all of us. And we have Andrienne Doyden, and she's going to be exploring the role of sustainable housing and how we can think about developing a more livable and resilient future. So there'll be a live event page. You can register. I hope you can come and join us for that as well. So thanks for coming this evening, and we're hosting a reception outside the room, so we invite you to come. I know some of you didn't get your questions answered, so please engage with Carolyn. And I just once again want to thank you for joining us this evening. And for those of you online, I want to thank you very, very much as well. And I hope you have a great rest of your evening. So one more time, please join me in thanking Carolyn.